The overall goal of this procedure is to PCR amplify retroviral integration sites from bulk genomic DNA and sequence the resulting DNA library, such that precise integration locations can be mapped to a reference genome.
This method can help answer key questions in basic and clinical retrovirology by analyzing the distribution of integration sites throughout a host genome. The main advantage of this technique is the DNA sequencing depth and the fact that many samples may be multiplexed in one sequencing run.
One day prior to transfection, plate 3.3 × 10⁶ HEC293 T cells in 10 milliliters of supplemented Dolbeco's modified Eagles medium in each of 5 100 millimeter dishes. The next day, use commercially available transfection reagents, or calcium phosphate, to transfect the cells with 10 micrograms of plasmid-carrying full-length retroviral molecular clones, or 9 micrograms of envelope-deleted single-round vectors, plus 1 microgram of a VSV-G expression construct. Add the mixture to the cell media dropwise. Following transfection, incubate the cells at 37 degrees Celsius in a humidified cell culture incubator with 5% carbon dioxide. After approximately 48 hours, harvest the virus-containing cell media using a volumetric pipette and pass it through a 0.45 micron filter by gravity flow. Next, concentrate the virus by ultracentrifugation at 200,000 Gs for 1 hour at 4 degrees Celsius. Following centrifugation, remove the supernatant until approximately 500 microliters of DMEM FPS remains, and